Hey guys, welcome back to One Stop Biology. So today we are going to start with a very important chapter. We are going to start genetics basically, and the very first chapter of the unit genetics, which is principle of inheritance and variation. Guys, I'll repeat it again. It is very important from boards and need point of view both. So you'll have to understand each and every three things starting from the basics to whatever I'm teaching here very well. And if you have any doubt, please do not forget to leave a comment on the same video, or you can also ask your doubts through the WhatsApp number which is given in the description of the video. Okay, so let's start with principles of inheritance and variation. We'll first see Mendel's law of inheritance, and then in this in this video we are going to study inheritance of one gene as well. So as we all know that Gregor Mendel, which is considered as the father of genetics, right? So Gregor Mendel is known as the father of genetics. So what he did is he conducted various experiments on the garden peas. Now before we study all this, do you know what exactly is genetics? Do you know what do you mean? What do we mean by genetics? So basically there is a branch of biology which is known as genetics, and it deals with inheritance. So it deals with inheritance as well as variation of characters from parents to offspring. So basically, we know that no two individuals are alike, right? So the inheritance and variation of characters from the parents to the offspring, basically, the study of this is known as genetics. Now, we know that inheritance is the process by which characters are passed from one generation to the other, and that is the basis of heredity, right? And this variation is basically the degree by which a progeny differs from their parents, right? So Gregor Mendel started studying genetics in pea plant, and he conducted hybridization experiments on garden peas for 7 years, 7 long years, which is from 1856 to 1863. And then he proposed the laws of inheritance in living organisms, right? So basically, during this Mendel's investigation into inheritance patterns, it was the first time that statistical analysis and mathematical logic both were applied to problems in biology. So basically, his experiments had a very large sampling size, which gave credibility to the data that he collected, right? And then also the confirmation of his inferences from experiments on successive generations of his test plants also proved that his results pointed to the general rule of inheritance rather than being just an unsubstantiated idea, right? So he did things like you know, conducted experiments on various traits of a pea plant, and these various traits, so each trait has basically two opposite sides. So if we see that in the picture below, we'll see that. So basically, what he did is there are always two opposite traits to every character. So if say a plant is tall, the other trait of it will be dwarf. If the seeds are yellow, the other trait of those seeds will be being green seed, right? So what he did is he conducted artificial pollination and cross pollination experiments using several true breeding pea lines. Now, what is a true breeding pea line? A true breeding line is one which has undergone continuous self-pollination. So it is true breeding, and it shows stable trait inheritance and expression for several generations. because it has always undergone self-pollination. So Mendel, what he did is he selected 14 true breeding pea plant variations, right? And these are in pairs. So basically these 14 traits were all this. So the first was seed shape. Now seed shape can be round or wrinkled. In the same way, seed color can be yellow or green. Flower can be violet or white. Pod shape can be full all or constricted. The pea pod color can be green or yellow. And the flower position can be axial or terminal. In the same way, we have the stem height as well. So stem of a plant can be either tall or dwarf. So these are various variations, right? These are various variations that he considered in all of his experiments. Now here you can see that these are the characters, right? So these are the characters. Here they have mentioned it is either dominant trait or recessive trait. So we'll talk about the dominant and recessive trait as well, right? So let's see what exactly he meant by describing these pairs, right? So what he did is he conducted so many experiments and then he chose these 14 true breeding pea plants and then he came up with inheritance of one gene. Now what exactly is inheritance of one gene? So basically let us take it as through an example. So say Mendel crossed tall and dwarf pea plant. So again, one character, two different traits, tall being the dominant one and dwarf being the recessive one. So basically he crossed tall and dwarf pea plant to study inheritance of one gene. So this basically character has come from one gene. Now we all know the structure of a flower, right? So if I have to label this, this is the petal, this one is the stigma. Here we have anther, we have stamen, and we have carpel, right? So what he did is he collected the seeds which are produced as a result of this cross. So he crossed a tall and dwarf pea plant. He collected the seed which the plant which is a cross of tall and dwarf pea plant produced, and he grew them to generate plant of the first hybrid generation, right? So this is also known as filial one progeny or F1 generation, right? So basically this is what he did, right? So he took two plants, a male and a female. He crossed, so he transferred the pollen from one to another, as we know that these true breeding plants always self-pollinate, right? And then he took the seed and he grew the seed into a whole plant, and this is nothing but the filial one. This is the first generation, right? Which is also known as filial one or F1 generation, right? Now, what he did after this? So, what he did after this is Mendel, what he observed is that all the F1 progeny plants were tall like one of its parents and none were dwarf, although the other parent was a dwarf plant, right? So, what he did is he then self pollinated the tall F1 plant, the F1 progeny that he found, which were always tall, he self-pollinated the tall F1 and found that in the second generation, in the filial 2 generation, some of the offspring were dwarf, while remember again that the parent is tall. Now, this character was not seen in the F1 generation. So, what he did basically is he basically crossed a tall, so this is a tall plant and this is a dwarf plant, right? So he did a between a tall and dwarf, and the first gen that he got was always a tall plant. There was no dwarf plant here. But when he crossed the tall plant with itself through self pollination, he got some tall and some dwarf as well, although there is no dwarf plant in the parent of these plants, right? So, based on these observations, what he proposed? Based on these observations, he proposed that something is there which was stably passed down unchanged from the parent to the offspring through the gametes over successive generations. Now, he called these things as factors, and those factors are now called as genes. Those factors are now called as genes, right? So here if you see the ratio of tall is to dwarf, right? What he studied that only one of the parental trait was expressed in the F1 generation, while in the F2 generation, both the traits were expressed, tall and dwarf, and that was in the proportion of 3 is to 1. 3 tall is to 1 dwarf, right? So this basically is because of genes which are passed from parents to offspring through the gametes over successive generations, right? So genes therefore are considered as the unit of inheritance. So genes are the unit of inheritance, and they contain the information that is required to express a particular trait in an organism. So basically, they are the things which contain information which is required to express a particular trait in an organism. Now, genes which code for a pair of contrasting traits. If there are genes which code for a pair of contrasting traits are known as alleles. Remember, they are known as alleles. So basically, they are slightly different forms of the same gene, right? So if we use alphabetical symbols for each gene, then the capital letter is used for the trait expressed at the F1 stage and small alphabet for the other traits. So say if we are talking about height, and if we say that capital T is for tall, then small t will be for dwarf, right? And these T and T are nothing but alleles of each other. These T and T are alleles of each other. Now in plants, the pair of alleles for height would be either both tall, one tall and one dwarf, or both dwarf allele, right? So remember this, that this will always be the pair of allelic pair of genes for height, right? Now Mendel also proposed that in a true breeding tall or dwarf pea variety, the allelic pair of genes for height are identical or homozygous. So it is either T or T, right? So both tall or both dwarf. This is known as identical or homozygous. Identical or homozygous, right? So these are basically, these two are genotype of the plant, right? These two are genotype of the plant, while the descriptive tall and dwarf are phenotype. These are phenotype of a plant. These are genotype So basically these are the genetic trait, these are the phenetic, these are the physical descriptions. So these are the phenotypes and these are the genotypes of the plant, right? Now, as Mendel found the phenotype of F1 heterozygote, which was one tall and one dwarf, right? And this basically is became the parent gene now, right? So let's see what he proposed later on. Let's see what he proposed after this. Yeah. So what he proposed that in a pair of dissimilar factors, as in the case of this plant. Here we have heterozygous genes, right? So in the pair of a heterozygous or dissimilar factors, one dominates the other, because what he did is he first crossed these, right? And what he got was this. But this generation what was not a mix of tall and dwarf. This generation phenotypically was tall itself, right? So that means that there is one factor which dominates the other, and that is why that factor is known as the dominant factor. That factor is known as dominant factor, right? And the other one is known as the recessive factor, right? So in this case, T is the dominant factor and T as in for tallness, and small t for dwarfness is the recessive factor, right? Now he observed identical behavior for all the characters that he studied, right? So what he did is he very logically and conveniently used capital and lowercase of an alphabetical symbol to remember the concept of dominance and recessiveness. Like we use capital T for tall and small t for dwarf. We do not use, say, capital T for dwarf and small d for dwarf. This will always be wrong, right? What he did is he used alphabetical symbol as in capital and lowercase for each and every factor and the both the alleles. So the dominant one will always have the capital factor, and the recessive one will always have the small case of the or the lower case of the alphabetical symbol, right? So alleles can be again similar as in the case of homozygotes. So if it is homozygote, it is this or this. If it is homozygote, if it is heterozygote, then it will be one this and one this. Why? Because the heterozygous plant is controlling genes of one character height. Now this kind of cross is known as the monohybrid cross. This kind of cross is known as the monohybrid cross. This is because in this kind of plant, it is just one gene which is controlling the character. Basically, it is one character which is controlling the plant. Now it can be height, it can be pea shape, it can be pea size, it can be plant color, or what all factors we studied, but just one factor of a kind, right? So from the observations that the recessive parental traits is expressed without any blending in the F2 generation, we can basically infer that when tall and dwarf plant produce gametes by the process of meiosis, the alleles of the parental pair they separate or segregate from each other, and only one allele is transmitted to a gamete, right? So let's see basically what it means through the diagram itself. So what he said is that we saw that when the basically when a plant produces gametes by the process of meiosis, only one allele goes to each of the gamete, right? Now this segregation of allele can be a random process, and there is a 50% chance of a gamete containing either allele, which is basically verified through the results of crossing. Now in this way, gametes of a tall, say TT plant, gametes of a tall TT plant have the allele T, right, and T, and gamete of a small TT plant have the allele small t and small t, right? Now during fertilization, the two alleles, which is T from one and T from this one, right, these are united, right, and these produce zygote, which has one capital and one small t. So basically these plants are producing hybrids which have these two alleles, one from the tall plant and one from the dwarf plant. Now since these hybrids contain alleles which express contrasting traits, the plants are known as heterozygous. We have studied it various times, right? These are known as heterozygous. Now, production of gametes by the parents and the formation of zygote. Then the F1 and F2 plants. All these can be understood through a diagram which is known as Punnett Square or Punnett Square, right? So this Punnett Square was developed by a British geneticist. Is developed by a British geneticist, Reginald C. Punnett. Right. So this Punnett square is nothing but a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. Now the possible gametes are written on two sides, usually on the top row and left columns, and all possible combinations are represented in the box below in the square, right? So this basically generates a square output form. So here if you see in this particular diagram, here it shows that the parental tall, which is a male plant, and the second parental plant, which is a dwarf female. So remember that this is the male plant, this is the female plant, right? Now these two basically are crossed and they produce gametes. The tall one produces two tall one gametes and the dwarf one two small t for dwarf ones. Now the generation that you get, so what we are doing here is if we basically use this and this gamete, we are getting this, right? So basically what we are doing, we are crossing the, we are basically checking all possible combinations here. So this with this, right? And then this with this, again this with this, and this with this, right? This is the combination that you can go for, right? That either of these and either of these can cross with both of them, and they'll form all heterozygous gametes, zygote, right? They'll form heterozygous zygote. So basically now what happens is that all the F1 plant of the genotype, when we now self pollinate this, so what we are doing is now we are using two of these plants, right? So again one with now heterozygous alleles and again a heterozygous alleles. The next Punnett square that you will see will be this, right? So now what we are doing is we are again separating both the gametes. So here we have one tall and one dwarf. In the similar way, since it is self-crossing, again we have one tall and one dwarf, right? Now when I cross this, either it will combine with the tall one of the other plant or the dwarf one of the other plant, right? So if it combines with the tall one, it will form a tall plant. If it combines with the dwarf one, it will form the tall plant but heterozygous one. Again, the other allele, if it combines with the tall one, it will be forming a tall but heterozygous. But if this combines with the dwarf, so basically we are combining a dwarf with a dwarf, it will form a dwarf plant, and that is why in the F2 generation, if in the filial 2 progeny, we see one dwarf plant. So now here if we see the phenotypic ratio, it is tall is to dwarf, right? And if we see the genotypic ratio, it is one homozygous tall, two heterozygous tall, right? And one homozygous dwarf. So the genotypic ratio becomes 1 is to 2 is to 1. Now, if I talk about the tall is to dwarf one, the phenotypic ratio becomes 3 is to 1 because there are 3 tall and 1 dwarf. So this is how we use a Punnett square, and we find out what exactly is the combination of the gametes and what kind of plants they are going to produce in the coming generation, right? Right. So if we talk more about the Punnett square now, basically from Punnett square we saw that 1/4 lead to a homozygous tall, half of the total progeny led to a heterozygous tall, and then 1/4 led to a homozygous dwarf, right? Now, though the genotype is this, but the phenotype is tall. Remember this. So, if we talk about the phenotype, 3/4 of the plants are tall and only one pair, which is both small t, are dwarf, right? So basically the characters T or tall here is dominant over the other allele, which is small t or dwarf, right? So this leads to a phenomenon of ratio which is 3 is to 4 and 1 is to 4. So I wrote that phenotypically the ratio is 3 is to 1 for tall is to dwarf, right? And genotypically it is, because we're talking about different now genes, it is either this or this or this. So these are three types of genotypes, and the combination is one of this, so 1/4 of this, half of this, and 1/4 of this, right? So the ratio is 1 is to 2 is to 1. So I hope you'll remember this very well. It is very important. I'll repeat it again that the inheritance of one gene is very important, right? So a cross of an organism showing a dominant phenotype with a recessive parent is known as a test cross. That is a test cross, right? So again, if you see here, this is a test cross wherein what we are doing is there is a homozygous recessive and a homozygous recessive here in here, right? Now, if we do not know what this is, and if we just know what these progeny are, we can basically derive what are the alleles of this, and we can derive what the plant is, right? So if I tell you that there is a parent which is homozygous recessive, that is say TT, and I don't know what the other parent is. But if I tell you the cross, there are four progeny, right? One is this, so basically one is this, the other one again is this, the other one again is this, and the other one again is this. Can you tell me what exactly is the other parent? You will have to just separate the gametes. So if we know that the one parent has both the recessive gametes, so here it will always be both the recessive gametes. Now to get this, right? I'll just clear it out and write it again. So to get this, what I'll have to do is I'll have to basically cross it with a tall gene. Again here tall gene. So if I cross it with a tall gene, again this will be there, this will be there. I'll cross it with this and I'll. So now I know that the other parent is a homozygous dominant one, right? Through this basically we can identify one of the parent as well if we know what is the genotype of the progeny, right? So this basically is known as the test cross. So guys, with this we have finished the inheritance of one gene. Now in the next video again we are going to cover some very important laws of genetics, which are law of dominance and law of segregation. And I hope you understood this video very well, and I would tell you again to ask me any small doubts if you have, rather it is, even if it's small or big. I'll even repeat this topic again by coming live on the YouTube channel. I would just request you again to ask me all the doubts that you have. You can either ask me doubts by just mentioning it in the comment of the video, or you can also just message me on the WhatsApp number which is given in the description of the video. I hope you understood the video very well. If yes, then please do not forget to like the video, and I hope you would have subscribed to the channel by now. Thanks guys. Bye bye.